Welcome back to Catalyst University. My name is Kevin Tokoph. Please make sure to like this video and subscribe to my channel for future videos and notifications. The topic of this video is going to be ubiquitination and the ubiquitin ligase system. And I'm probably going to put this video both in the prion playlist and also the protein metabolism playlist. And it turns out that if you have a way to synthesize something in the human body, there should also be a way to get rid of it. That's an important thing. What comes in has to come out. And so proteins are obviously synthesized by the ribosome, but what happens when you don't need that protein anymore? So perhaps the protein, you don't need its function, you need to get rid of it. Or if the protein becomes damaged, oxidized by free radicals, so on and so forth, you need a way to get rid of the protein. And it turns out that one method of doing that is called ubiquitination. What ubiquitination is it's the attachment of very large proteins, or peptides I should say, called ubiquitin to a protein, and that actually signals its destruction. This UB right here in this diagram represents ubiquitin, and it's approximately 76 amino acid residues long. It's a very large peptide, and it's very well conserved between species. If you have a protein that ends up with ubiquitin residues attached to it, it will ultimately be targeted to a proteasome, which is the structure right here, basically a molecular garbage can. Really, it's more of an incinerator, but we'll talk about that on the next slide. First, let's talk about the ubiquitin ligase system and talk about how these ubiquitin proteins or peptides are attached onto a protein that needs to be destroyed. So the ubiquitin ligase system is a system of three enzymes denoted E1, E2, and E3. So E1 has a thiol group on it right here from a cysteine, and we need to get this ubiquitin onto it. In fact, the ubiquitin residue that actually gets directly attached, I believe, is a glutamic acid or glutamate residue. And the first reaction is a synthetase reaction. It uses the power of ATP to form a thioester bond between enzyme 1, its cysteine residue, and this ubiquitin carboxyl. Okay, so you have a thioester group right here. That's the initial attachment of this ubiquitin group. Now you actually have to attach the ubiquitin to the E2. So this is actually called a transubiquitination reaction because you're actually going to directly move the ubiquitin from E1 to E2. Okay? So this is just a simple nucleophilic acyl substitution reaction. Essentially, this ubiquitin is going to be moved onto this E2 through the nucleophilic attack by this E2 thiol, again a cysteine residue, and now the ubiquitin is attached to this E2. All right. We're going to proceed here in this direction. This E2 with the attached ubiquitin is going to be picked up by E3. Now the purpose of E3 is twofold. One, it's going to actually pick up this E2 with bound ubiquitin, but it's also on its other side going to bind the substrate. And in this case, the substrate is going to be the protein that needs to be degraded. Typically, the ubiquitins are attached to lysine residues on the protein. Okay, so in other words, what E3 is going to do, this is actually the ubiquitin ligase, it's actually going to attach this ubiquitin onto this amine of the lysine R group. And so now what we can say is the ubiquitin is now an amide or a peptide bond to the protein that needs to be degraded, labeled as the substrate here. Okay? Now, after this step is over with, the E2 is now empty. It will then leave the enzyme and then go back to E1 to pick up another ubiquitin. So the purpose of E2, it's going to pick up the ubiquitins from E1 and then bring them to E3. And the reason that's important is because you're not just going to ubiquitinate this protein one time, you're usually going to do it many times. Here it shows 4 times, but it can be even much more than that. And so what's going to happen is, again, this protein is still going to be bound to E3. It will pick up another fresh E2 with bound ubiquitin, and you'll just repeat this cycle several times, okay, and attach a bunch of ubiquitins to that protein in the process. And so in the end, you're going to have this damaged protein, or it could just be a protein that's used up its function, the cell doesn't need it anymore. You're going to have a bunch of ubiquitin molecules attached to it, okay? These ubiquitin molecules are going to signal to the cell that this protein needs to be destroyed. And the way it will be destroyed is through this very large macromolecular complex called the proteasome, sometimes called the 26S proteasome, very large. The best way to think about it is it's a molecular garbage can. I usually think about it as an incinerator as well. In fact, if you actually look at its chemical structure, it looks like a garbage can, except for it has an opening on both the bottom here and the top, however you want to call that. And so what will literally happen is one of these tops will actually flip open, just like when you have those trash cans, you push the pedal and it opens. Well, it will actually open, and this opening right here will actually recognize these ubiquitin pieces. Okay, that will trigger initial binding to the proteasome, and from there you're going to have this protein move into this tube. Okay, in which case there is a trypsin-like activity that will ultimately hydrolyze the protein into fragments like this, which will then be recycled. The reaction itself requires ATP, but not in the proteolysis of this protein. The ATP is actually just necessary for subunit binding of the proteasome, very similar to the way ribosomes require nucleotide triphosphates when the ribosomes assemble. Okay, very similar. Okay, and in fact, the mechanism, which we'll look at in a separate video of the degradation of the protein, is actually a threonine hydrolysis. It's a threonine protease, very similar to a serine protease that we looked at in enzyme mechanisms a long time ago. In addition to the ubiquitin binding to this region of the flap right here, as you can see in another diagram down here, there are some amino acid residues that have to have some degree of complementary binding to the proteasome. And we've zoomed in on some of them right here. What we can see is there's actually a consensus sequence that's a complete match and will give you very tight binding between the protein that needs to be degraded, here it's in yellow, and the proteasome. That will induce very quick and rapid degradation. You can also have more complex sequences where there's a little bit of overlap, but not as much as the exact consensus sequence. That will still lead to protein degradation. There's also what's called a bias sequence. This is where there's not many matches at all. You can see here the only match is this red area. The binding is below threshold, and these proteins will be the most difficult to degrade from the proteasome's perspective. Okay. In other cases, you can have complex sequences where the sequence is really just flexible, in which case you can kind of imagine the protein just kind of fluttering and slithering in. It easily fits into the degradation channel inside this proteasome, making it very easy for the proteasome to degrade it. But also you can have other bias sequence where you have a compact protein that's not flexible and it does not insert easily into that degradation channel of the proteasome, again making those proteins much more difficult to degrade. And sometimes there are actually non-proteasome mechanisms to also degrade the protein. But this is the major predominant mechanism. Okay, so again, you really have to have two things occur. You have to have this ubiquitination, but you also have to force this protein that needs to be degraded into this hollow tubing, into the channel of this proteasome. And as you can see here in this diagram, these scissors represent where the proteolysis actually occurs. So you can see it goes into the tube itself, deep in there, and then it's a controlled proteolytic environment, which is catalyzed by a threonine residue within the active site. Very similar in mechanism to the serine proteases that we saw much earlier in biochemistry. And this will ultimately, as you can see here, lead to the complete destruction of that protein. And these peptides and amino acids can then be recycled for other purposes. All right. Please make sure to like this video and subscribe to my channel for future videos and notifications. Hopefully you learned something in this video, and I hope you enjoyed it. Thank you. Join us next time.